I hope that's clear. It's 10:35 now, so we can start. So from today, you're starting a series of lectures on bacteriology. And after bacteriology, you'll have mycology. Bacteriology is the study of bacteria, as the name suggests. Well, mycology is the study of fungi. Of course, these are medically important fungi. So that's a series of lectures you'll have until the end of the academic year. So today we look at introduction to bacteriology. We'll also look at bacterial anatomy and bacterial physiology. And this is, class rep, are you recording? Shall I?
Yes, we are.
Okay. So let's first look at introduction to bacteriology. As I was saying, these are foundation lectures that will be important for your understanding of bacteriology. It's usually a very tight program for your program. So you need to really understand the foundations of bacteriology. So bacteriology, as I had said, is a study of bacteria. And what is of interest to us is medical bacteriology, which is a study of bacteria that cause disease in humans. What you need to remember is that bacteria are too small, cannot be seen by the naked eye. And so we use microscopes to view these bacteria. And the microscopes that you will be using is a light microscope. So in the light microscope, we look at the bacterial cells. The shape is usually very important in giving us a clue as to what bacteria we are dealing with, the size, the arrangement, and various characteristics. For example, is this bacteria motile? That is, does it have the ability to move? Not all bacteria are motile. Do they have a capsule, etc.? And then we have the special microscopes, which are only used, as the name suggests, under special circumstances. For example, the phase contrast and dark field microscopes, which are for the visualization of refractive and slender organisms, because the light microscope uses light. And so some of these organisms are very refractive so that you're not able to see them under a light microscope. They're also very slender, so you'll not be able to see them in the light microscope. You need a dark field so that you're able to see the bacteria. Then you have the immunofluorescent microscope. An example is what you're now commonly using in the diagnosis of Mycobacterium tuberculosis, which causes TB. And of course, you have the electron microscope. You will not come across it during routine labs, but you use it to look at the fine structures of bacteria for higher resolution, to look at the nucleus and so on and so forth. So these are diagrams showing to the left, the light microscope. You're already familiar with the light microscope. It's important to know the parts of the light microscope and also the function of each of the sections of the parts of the microscope. In the middle is a dark field microscopy. So as the name suggests, you have a dark field so that you're able to see the very refractive bacteria. This is an example of Treponema, a genus of bacteria referred to as Treponema. Treponema pallidum, subspecies pallidum, which causes syphilis. To the right is an electron microscope, which we use to look at the finer features, the finer structures of bacteria. So the unstained bacterial cells are refractive, and so you're not able to visualize them clearly under the light microscope. And so another very important thing in bacteriology is the staining of these bacteria. The staining gives them color, and also important because it differentiates special characteristics, because different species of bacteria with different characteristics stain differently. Another thing to remember is a bacteria is slightly negatively charged at a neutral pH. So we use this sometimes to use special staining techniques. For example, you may want to stain the background, in which case you use a a negative stain so that it's repelled by the bacteria. The bacteria will not be stained, but the background will be stained. So you'll see it like a refractive cell. And of course, the opposite, the positive stains then will be attracted to the bacteria and are going to stain the bacterial cell itself and not the background. So what are the types of staining, of the stains that we have? So you can have the simple stains or the differential stains. In simple staining, you only use one type of a stain. So you have a positive stain, or you can have a negative stain. As I had explained, bacteria are negatively charged at a neutral pH. So a positive stain is going to be attracted to the bacterial cell and stain it, while a negative stain is going to be repelled by the bacterial cell and stain the background. And then you have the differential stains where you use more than one stain in different stages. What you're going to be hearing a lot is a gram stain. So you'll hear gram-positive bacteria, gram-negative bacteria, and so on. So that's a differential stain where you use more than one stain. So this is an example of gram staining. So we use different stains, and the stains that we use will be attached or will be attached to the bacterial cell depending on the characteristics of the bacterial cell. We have 4 steps in gram staining. So we have the primary stain, and we have the second stain, which is iodine, and then the third step is decolorization using alcohol, and then we have the counter stain, which is the last step. So in the counter stain, what you'll be using in the lab is neutral red. I think what we have is neutral red. So depending on the characteristics of the bacterial cell, which you're going to look at later, the bacterium can either take the primary stain, in which case it remains purple, and these are the gram-positive bacteria. So to the left of the slide, you can see gram-positive cocci. Cocci are round, they appear purple. At the bottom right, we have gram-positive bacilli. We're going to look at the shapes later, but look at the color now. So these gram-positive bacteria take on the primary stain to remain purple bluish. And the gram-negative bacteria do not take on the primary stain because they're easy to decolorize due to some characteristics in their bacterial cell. And so they take the counter stain, which is the last step. In this case, we used neutral red. So they'll take up the color red or pink. So to the right, we have the gram-negative bacilli, and at the bottom left, we have the gram-negative cocci. What you need to remember at this stage are the colors of the gram-positive bacteria and the gram-negative bacteria and what they appear like on a light microscope. So this is using a light microscope. Another very important thing that we do in bacteriology is what we refer to as culture, growth, and identification of bacteria. Culture simply means the isolation of the bacterium from patient sample. So you have a patient with pneumonia. How do you know what bacteria? There are many bacteria that can cause pneumonia. How do you know which of these bacteria is causing the pneumonia? So you need to isolate the bacterium in vitro, which means outside the body. So it involves the growth of bacteria in vitro outside the body from specimen from patients. Why do we do this? To identify, and we refer to this as laboratory diagnosis. So when we talk about laboratory diagnosis, it is a process of isolating the organism that is causing a certain infection. So you take a specimen from a patient and then you're going to isolate it and identify it. So which bacterium is it? Is it gram-positive? Is it gram-negative? And for this, we use artificial culture media. So the artificial culture media, we try to, in as much as possible, to mimic the environment in the body under which bacteria grow. So how do we identify? It's based on the phenotypic characteristics. Phenotypic means, you already know what phenotypic means as opposed to genotypic. So the phenotypic is the outward appearance or the outward characteristics. The genotypic characteristics, on the other hand, are what's the genome. So we can also identify bacteria using their genome. So those are the genotypic methods. But for the phenotypic characteristics, we are interested in the microscopic morphology. As I said, we'll use the light microscope. So what is the shape of this bacteria? Is it round, which we refer to as cocci, or is it a bacillus? Is it gram-positive or gram-negative? So that's the staining properties. If you use a gram stain, is it gram-positive or gram-negative? And then we isolate using artificial culture media. And once they grow on the artificial culture media, then we describe the cultural characteristics. So different bacteria will grow differently on the artificial culture media. So we are looking at, does it have smooth colonies? So as it grows, we describe that growth as colonies. So the growth is colonies. Are they smooth colonies? Are they rough colonies? Are they large? Are they small? What's the color of the growth on artificial media? And then the next step usually is to do biochemical tests and reactions, because you may not be able to identify using the cultural characteristics. And so we use biochemical tests and reactions. So as you're going to be looking at specific bacteria, you're also going to be describing the biochemical characteristics. Some of the tests that we use are catalase. So is this bacterium a catalase producer? Does it produce another enzyme, which is important, called coagulase? So those are the tests that you're going to be doing in biochemical tests. And then we also will do typing, which are immunological tests. So some bacteria have different strains. You want to know, for example, in cases of, say, an outbreak of pneumonia, you want to know this bacterium, one of the commonest causes of pneumonia is Streptococcus pneumoniae. You want to find out what strain of this bacterium is causing this disease. And that's where typing comes in. It is looking at the antigens that are found on the bacterial cell. And of course, you can also identify using DNA characteristics, looking at the genome or parts of the genome of the bacterium. You have heard of PCR tests. You have heard of sequencing of the entire genome of the bacteria. So you can also use those for identification, but these tend to be more expensive. So what you mostly do is the isolation on artificial culture media. And also for those that are difficult to isolate, there's some that are very exacting in their environmental requirements. So we use immunological tests. So indirect methods of detection, we've talked about this. Genetic components, an example is use of gene probes. So you target a certain part of the bacterial genome. You also have the immunological method, so you can either target or look for the antibody against the bacterium, or you can look for the antigen, the bacterial antigen, whether it's present in the person with the infection. So there's the international classification of bacteria. We also refer to it as binomial nomenclature. This is universal even for other organisms. So this is what we use most of the time, all of the time, to refer to bacteria. You already know this from basic biology, that genus, we have the genus, which is the first part of the scientific name of an organism. And you have the second part, which is a species. So the way we write it is as the genus, the first letter should always be capitalized. And for the species name, we write it using the lowercase. That's why it's called the international classification, so that anybody around the world, when you write it, will be able to know what you're talking about. So when typing, then this should be italicized or underlined, like the example. there. So one of the bacteria that you first learn about is Staphylococcus aureus. So the first one is italicized. The second one is not italicized, but it's underlined. So either of these are internationally recognized ways of naming bacteria. So how then do we classify bacteria? And these bacteria can be classified by the macroscopic appearance, and this is the colonial morphology. So remember we said that we grow them on artificial culture media, and different bacteria have different characteristics on these artificial media, which give us a hint of what bacterium you would be dealing with. Then the microscopic appearance. So you're looking at it. What you'll mostly be using is the light microscope. What is the shape of the bacteria? What are the staining characteristics? And the gram stain is what we'll be referring mostly to. Then what are the characteristics of growth and metabolic activities? Oxygen use is very important. Some bacteria will not survive in oxygen. Others require oxygen. Others require reduced oxygen concentration, and so on and so forth. And we also look at some utilization of different things like carbohydrates. So carbohydrate utilization also important in classification of some bacteria. We've talked about antigenicity when you're using immunological tests, and also the genome characteristics if you're using genotypic methods. An important part of interest to the pharmacists is in the cases of antimicrobial resistance. We constantly look for resistant genes in the genome of these bacteria to know exactly what is causing the bacteria to be resistant to the commonly available antimicrobial agents or drugs. So let's go straight to the classification of medically important bacteria, because this is medical bacteriology. So we've already talked about the important characteristics, one of which is the shape. So you can describe bacteria as being cocci, that's plural, singular is coccus. So cocci are spherical in shape. They can also be bacilli or rods. So you can either call them rods or bacilli. They can be spiral, like the example we saw of the dark field microscopic view of Treponema species. And they can also have a combination of shapes. So when you look at the microscope, you're not able to tell, is this a bacillus, is this a cocci? They seem to have all those, and we refer to these as leiomorphic bacteria, having a leiomorphic shape or being coccobacilli. So they are not predominantly cocci or bacilli. They seem to have both of these. So we're still talking about classification. In addition to the shape, the gram stain is very important. Bacteria can be gram positive or gram negative, but there are those whose cell wall characteristics do not allow us to be able to classify them as either positive or negative. We refer to these as stain variable, sometimes stain indeterminate, in which case we use special stains other than the gram stain to stain them. So what we commonly use is a combination of the two, the shape of the bacterium and the gram stain reaction of the bacterium. So you refer to this bacterium when you're talking about a certain bacterium, you need to say these are gram positive cocci or gram positive bacilli or gram negative bacilli or gram negative cocci. So that's what you'll be hearing a lot of. So morphology, the other one is based on morphology, and this is an important special stain, the ZN stain. ZN stands for the Zil-Nielsen stain. Now there's some bacteria which in their cell wall are very rich in mycolic acids, which are lipids. An example is Mycobacterium tuberculosis, which causes TB. It's very rich in lipids. And so it's not able to take up the gram stain. So we use special stains, and they're also acid-fast bacilli. So even if you decolorize using acid, it's not going to be decolorized. So we use a ZN stain, Can also use cellular characteristics. Some bacteria have capsules and others have flagella, which help them to move, so they're motile. We talked about the atmospheric requirements, which we shall look at more detailed under the bacterial physiology. So is this bacterium able to grow in presence or absence of oxygen? In which case, we divide them into two. Aerobic bacteria grow in the presence of oxygen, where anaerobic bacteria grow in complete or not complete. For the strict anaerobes, then they require complete absence of oxygen. We can also classify based on other growth requirements of various temperatures. So most medically important bacteria will grow in the range of the body temperature from 35 to 37 degrees centigrade. But we have those that are able to grow at higher temperatures and also at lower temperatures. And of course, the ability to cause disease. So we divide these into two. The pathogenic, these are bacteria that have the ability to cause disease. And you have the non-pathogenic, which even if you find them, you're not too worried because they're not pathogenic unless under special circumstances such as immune suppression. So let's look at the anatomy of the bacterial cell. So bacteria are prokaryotes, and these are organisms without a membrane-bound nucleus. So we refer to that part of the bacteria which has the genetic material as a nucleoid. So you're going to see that later. And then you have what we refer to as a protoplast, which contains the nucleoid and anything else, the cytoplasm. And this is bound by a thin, elastic, and semi-permeable cytoplasmic membrane. The cytoplasmic membrane similar to eukaryotes, so it's a phospholipid bilayer. And closely covering the cytoplasmic membrane is the cell wall. And some bacteria also have a capsule covering the cell wall. So what is a cytoplasm? You already know this from physiology. Cytoplasm is aqueous environment packed with ribosomes and other complexes. Excuse me a minute. So we've already covered that introduction. So remember that bacteria are prokaryotes. They have a nucleoid. So you should not at any point talk about the bacterial nucleus because it doesn't exist. It has a nucleoid. So we already talked about the shapes, but this has diagrams showing us the different shapes. So we have two basic shapes. We have the coccus, that's a singular, which are spherical, and we have the bacillus, which are rod-shaped. So rod-shaped bacteria, the bacilli, can also have different shapes. We have the comma-shaped, for example, bacteria in the genus Vibrio. And you also have the spiral-shaped bacilli. So for example, Spirillum and spirochetes. An example of a spirochetes is a bacterium that causes syphilis. And you also have the filamentous bacteria. So this shows the different shapes, the coccus. We have the coccobacillus, so it's neither a coccus nor a bacillus, too small to be a bacillus. So we refer to them as the coccobacillus or the leiomorphic bacteria. This diagram shows a closer look at the bacterial cell structure. So from the inside, you have the nucleoid, and bacteria have circular DNA. Then you have the cytoplasm, and you have the plasma membrane or the cytoplasmic membrane. And you have the cell wall. And for those bacteria that have a capsule, the capsule will be above the cell wall or covering the cell wall. And for those that are motile, they have flagella or flagellum, singular. So let's look at the bacterial nucleoid. Bacteria have a single circular double-stranded DNA, most bacteria. Some may have double, but most have single circular double-stranded DNA and contains most of the genetic information of a bacterial cell. Now, because the bacterial cell is so small, the DNA has to exist in a supercoiled state to be able to fit into the bacterial cell. Now, some bacteria may also contain what's referred to as the extra chromosomal DNA. So the DNA that we find in a nucleoid, we refer to that as a chromosomal DNA, but bacteria may also contain DNA material outside of the chromosomal DNA, which we refer to as extra chromosomal DNA. And these are referred to as plasmids. So this extra chromosomal DNA is found in plasmids. Then we have the ribosomes. We already know about the ribosomes from physiology. So these ones are smaller, 10 to 20 nanometers, and they have a sedimentation coefficient of 70S. Remember from eukaryotes, of which human cells are, they have a sedimentation coefficient of 80S. Now the bacterial ribosomes have two subunits, the 30S and the 50S subunits. Of course, they're numerous in growing cells because they are for protein synthesis. And sometimes you may be able to see polysomes, which are multiple ribosomes attached to single messenger RNA molecules during vigorous protein synthesis. Of course, you have the cytoplasmic membrane or the plasma membrane, which is thin and elastic. It's a phospholipid bilayer, so it consists of phospholipids and proteins. What are the functions of the cytoplasmic membrane? It forms a permeability barrier. It's important for the transport of solids. It's important for energy generation, and you know from physiology that it's a location of numerous enzyme systems. Then we have the cell wall. The bacterial cell wall is very important and helps us to be able to divide bacteria into either gram-positive or gram-negative bacteria. Not all bacteria have cell walls. There's a special genus of bacteria referred to as mycoplasma, which do not have a cell wall. So the cell wall encases the protoplast. The protoplast is whatever is enclosed by the cytoplasmic membrane. It's strong and relatively rigid, but also has some elasticity to give the bacteria its shape. It's openly porous, 2 solute molecule less than 10 kilodalton, and a mass of less than in mass and less than 1 nanometer in diameter. Important because it supports the cytoplasmic membrane against high internal osmotic pressure. And it's very important. The integrity of the cell wall is what is important for cell survival. And you'll see later when you learn about the antimicrobial agents or the drugs against bacteria, is that a majority of them target the cell wall. Once you interfere with the integrity of the cell wall, then you kill the bacterial cell. And that is a process referred to as lysis. So the process of lethal disintegration and dissolution is referred to as lysis. The cell wall also plays an important part in cell division. And what you need to remember in bacteria is that the main strengthening component is referred to as a peptidoglycan. So you'll find us referring to it as PG. So peptidoglycan is what you need to remember. Now we are seeing one important difference between gram-positive bacteria and gram-negative bacteria. Gram-positive bacteria have a thick peptidoglycan layer. Your gram-negative bacteria have a thin peptidoglycan layer. Now, the peptidoglycan of both gram-positive and gram-negative bacteria is composed of two important acid molecules, NAG and NAM, which are linked alternatively. NAG stands for N-acetylglucosamine, while NAM stands for N-acetylmuramic acid. So these are linked alternatively. And the strands of NAG and NAM are then connected by interpeptide bridges. And these bridges are what make the peptidoglycan layer so strong. So N-acetylmuramic acid also carries a short peptide, which contains these amino acids. So sometimes they change. Sometimes it changes depending on the bacteria that you're dealing with, but generally it's L-alanine, D-glutamic acid, mesodiaminopimelic acid in gram-negative or L-lysine in gram-positives. Then you have the D-alanyl, D-alanine. And this is where at the D-alanyl, D-alanine, the interpeptide bridges are found. So different bridges, different strands, so making a very strong peptidoglycan layer. And this is a diagram showing the basic building blocks of the bacterial cell wall. Remember, we have two acid molecules, NAM and NAG. NAM, remember, carries a peptide bridge. Then you can see the interlinkage at the D-alanine, D-alanyl, D-alanine. That's where the interlinkage occurs. And you can see to the right a peptidoglycan unit of the two NAM, NAG, and the peptides. So sometimes the amino acids will change depending on the bacteria. So we are still talking about the cell wall. Another difference of the gram-positive and gram-negative bacteria is a gram-positive bacteria contain teichoic and lipoteichoic acid. Now, in addition, remember we said that gram-negatives have a thin peptidoglycan layer, and this is because they have an extra layer above or covering the peptidoglycan layer, and this is referred to as the outer membrane. The outer membrane contains a very important characteristic or component in gram-negative bacteria, which is referred to as LPS. LPS is lipopolysaccharide. Why is it so important? What are the functions of LPS? Now, the LPS is not, or the outer membrane is not freely permeable. Molecules must get in through porins. Porins are protein channels. So if they are too large, the molecules will not get into a gram-negative bacterial cell. And as you learn when you learn about the drugs that we use against bacteria, some drugs cannot be used against gram-negative bacteria because they are too large to pass through the porins. And then also, LPS is important because it acts as adhesins. Adhesins are important because they adhere onto a host cell, and this is usually the beginning step of establishing infection. Also important because it protects the peptidoglycan layer. Remember, it covers the peptidoglycan layer from the effects of lysozyme. From physiology, remember that lysozyme is found in saliva and other body fluids. And we've already talked about the inhibition of entry of antibiotics that are too large to pass through the porins. So gram-negative bacteria also have what's referred to as lipid A, which is a component of LPS. And this is important because it forms endotoxin. And endotoxin may give rise to endotoxic shock when released in the bloodstream. So when you start doing the rounds in the wards, you may hear about toxemia or endotoxic shock. So this is caused by the lipid A component of LPS. So in some bacteria, the peptidoglycan layer is covered or linked to mycolic acids. Mycolic acids are lipids, for example, in mycobacteria. And that is why we see that we do not use the gram stain if we are suspecting mycobacterium tuberculosis, for example, infection. We use special stains. So mycolic acids form an extremely hydrophobic external layer. So this diagram shows the differences between the gram-positive and the gram-negative bacterial cell wall. On the left is a gram-positive bacteria. Remember, we say it has a thick peptidoglycan layer. It also has teichoic acids and lipoteichoic acids. Lipoteichoic acids go all the way to the cytoplasmic membrane or the cell membrane. And then on the in gram-negative bacteria, they have a thin peptidoglycan layer. Then on top of the peptidoglycan layer, we have the outer membrane, which has porins and anthocyanins and so on. So only molecules that are able to pass through the porins can get into the gram-negative bacteria. So some bacteria on top of the cell wall have capsules. Capsules are mostly polysaccharides. Not all are polysaccharides, but most are polysaccharides. So they form what we refer to as a polysaccharide slime. Why is it important? Because it helps bacteria to evade host defenses, so it's not destroyed by the immune response. It also helps to adhere onto surfaces. So it's a virulence factor. You're going to talk about virulence in some different lecture, virulence and pathogenicity. So it's a virulence factor. It's anti-phagocytic. So to the right is an example of a negative stain. Remember, we said negative stains will be repelled by the bacterial cell. So it's going to stain the background, but not the bacteria. So in this case, the background is stained purple. And then we have the bacteria being seen. The bacteria are bacilli. So the dark bacilli, but there's also a large capsule. The white part is a capsule. So that's how we look for capsules in bacteria, or stain for capsules in bacteria. Then we have the flagella, which are important in locomotion. Not all bacteria have this. Flagella originate in the protoplasm, and they're anchored to the bacterial cell by what's referred to as a basal body. So there are different arrangements of flagella that can happen. So we have the monotrichous, where there is only one polar flagella. Examples are like those bacteria that cause cholera, the Vibrio cholerae. We have Lophotrochus, where we have tufts of flagella on both parts of the bacterial cell. We have Amphitrichus, where we have a polar flagellum on either part of the bacterial cell. Then you have the Peritrichus, where we have numerous flagella all around the bacterial cell. So these tend to be very motile. They move fast. So the other important parts of a bacterial cell are the fimbriae and the pili. Fimbriae are found peritrichously, which means that they are found all around the bacterial cell. And for those bacteria that have flagella, they are more numerous than the flagella. Why are they important? Because they mediate adhesion between the bacterium and the host cells. Now we have two kinds of pili. We have the sex pili and the common pili. The common pili are for attachment. Well, the sex pili are similar to the fimbriae but are longer, and they initiate the process of conjugation. Conjugation, as you learn when you look at bacterial genetics, is a process through which bacteria exchange genetic material, DNA material. So they use that using the sex pili. Then we have the bacterial spores. Not all bacteria produce spores. And when you're describing bacteria, usually we describe them like, is it capsulated? Does it form spores? Is it gram-positive? And so on. So for those bacteria that have spores, it gives them an advantage because spores are a highly resistant resting phase. So they do not proliferate or they do not divide when they're in this state, also referred to as the endospores. Important medically important bacteria that produce spores include bacteria in the genera Bacillus and Clostridium. Now, they help the organism to survive in dormant states. And these spores tend to be resistant to even most of the common disinfectants that we use. So the disinfectant can get rid of a bacterial cell, which we refer to as a vegetative cell, but will not be able to get rid of the spores. They're also resistant to drying and heating. And so to get rid of spores, we usually have to get to a high temperature and for a longer period of time to be able to kill the spores. So moist heat at 100 to 120 degrees centigrade or more for 10 to 20 minutes are needed to kill spores. Well, in comparison, you only need about a temperature of 60 degrees centigrade to kill vegetative cells. So vegetative cells are the bacterial cells that are active or are able to actively divide. Now, spores may remain viable for many years, and when the environmental conditions are conducive, then they undergo a process referred to as germination, which is the reversal of the spore. That is referred to as germination from the spore to the vegetative cell, and then they are able to continue dividing. So this is a diagram showing the endospores. So endospores can occur at different sites of the bacterial cell or of the vegetative cell. So they can be at the end, for example, in the middle one here, in which case this is a common one, we call it having the drumstick appearance. They can be subterminal or they can be in the middle. The other one shows a cross-section of the spore and the process of germination from the endospore to a new bacterium or a vegetative cell, which is then able to survive. So that's bacterial anatomy. I hope it is clear because it's important for your foundation in medical bacteriology. So we now look at bacterial physiology. What is bacterial physiology? You've already done physiology in first year. So you know that physiology is a specific means by which a particular organism obtains energy and raw materials to sustain its growth. So this is its nutritional type. What are the physical conditions it requires to be able to do that? And these two usually are what we use to reflect the fundamental physiological characteristics. The physiology is important because it helps us to mimic those conditions in the lab and be able to isolate the bacteria in the lab, which is very important for isolation. You want to say this case of meningitis was caused by this bacterium, or this case of a urinary tract infection was caused by this bacterium, which also then guides your treatment options. That is why isolation is so important. So the nutritional types, there are three basic features used to group organisms into nutritional groups. What is the energy source? What is the hydrogen donor? And what is carbon source? So the nutritional source, you already know this. Energy for ATP may be obtained from light. These are the phototrophic organisms. Through chemical oxidation, these are the chemotrophic organisms. What about the hydrogen donor type? The organotrophs require organic sources of hydrogen, while lithotrophs use inorganic sources of hydrogen. Then you have the carbon source. The autotrophs fix carbon dioxide directly, while the heterotrophs require an organic source of carbon. So usually we combine the energy and the hydrogen donor designation so that you have, for example, chemoorganotrophs, and these are the majority of the medically important organisms. And you have the chemolithotrophs and the others. But for us of interest, which is important for making artificial media, most medically important organisms are chemoorganotrophs. So you already know this. These are the physical conditions that are required for growth. All living organisms use oxidation to transfer energy. Remember the formula for oxidation, there's a molecule, there's a removal of hydrogen, then they need another molecule to receive the electrons. So in aerobic respiration, remember for bacteria, we say there are those that can survive in oxygen and there are those that cannot survive in the presence of oxygen. So for aerobic respiration, the final electron recipient is oxygen. For the anaerobic respiration, the final electron recipient is an organic molecule, and you know that we call this process fermentation. So this is from biochemistry. The physical conditions aerobic respiration results in the formation of highly toxic compounds. So remember reactive superoxide and also hydrogen peroxide. So bacteria that are able to survive in presence of oxygen, these are the aerobes, produce two very important enzymes, superoxide dismutase and catalase, and they catalyze these reactions that are shown down there. So superoxide dismutase to hydrogen peroxide, and then hydrogen peroxide to water and oxygen. So anaerobes, which are bacteria that cannot survive in the presence of oxygen, do not have these, and so they require complete absence of oxygen for you to be able to successfully isolate them in the lab. So based on the growth atmosphere, we then group bacteria according to their atmospheric requirements. So we have the strict or obligate aerobes, which require or can survive in ambient oxygen of 20%. Then we have the strict, we also refer to them as obligate anaerobe. So you can either say strict anaerobe or obligate anaerobe. These require complete absence of oxygen. So they do not have the two important enzymes that are required to survive in the presence of oxygen. Then we have the facultative anaerobes, which grow better in oxygen, but are still able to grow in its absence. We have the aerotolerant anaerobes, which are anaerobic but can tolerate exposure to oxygen, but not at high levels of 20%. Then we have the microaerophilic bacteria, which require reduced oxygen level. And we have the capnophilic bacteria, which require increased carbon dioxide levels. So these terms are important because you'll be hearing us say, for example, a bacterium Staphylococcus aureus is a facultative anaerobe. Clostridium tetani is a strict anaerobe. So you need to keep this in mind throughout bacteriology. So what about the growth temperature? You need to remember that the human temperature range is 35 to 37. And so most of the medically important bacteria will be what's referred to as mesophiles, which is the first group there. And these grow best at a temperature range of 20 to 40 degrees centigrade. Then we have the psychrophiles, which grow best at low temperatures. So refrigeration does not kill them. Then we have the thermophiles, which grow best at high temperatures of greater than 60 degrees centigrade. So most medically important bacteria are mesophiles because that's where the normal range of the human body temperature lies. So we have looked at the environmental characteristics. At the beginning, we talked about the importance of isolating the bacteria that is causing a certain infection. So how do we achieve this in the lab? How do you know when growth has occurred in the lab? So bacterial growth involves both an increase in the size of the organisms because they're taking in nutrients, but also an increase in their number. An increase in their number is what gives us the colonies that we look at and describe in artificial growth media. So we can look at bacterial growth in three main ways: development of colonies. So you're going to be talking about colonies a lot in bacteriology. And that is where you use what we refer to as solid artificial media. Now you can also use liquid artificial media. So for you to know that growth has occurred in liquid media, there has to be transformation of a clear broth. We always start with a clear broth to a turbid suspension. So when there's turbidity, you know that there's growth of organisms. And this is also why when you are working with drugs that are in liquid form, very important to make sure that they're clear. And also things like normal saline. Before we give them to patients, always make sure that they're clear. That's why expiry dates are important. But even if it's within the expiry date, it's important to make sure that it's clear, because bacteria can get in and grow in some of these drugs or normal saline. There's also what referred to as biofilm formation. And this is especially important in hospitalized patients who have indwelling catheters. It could be a urethral catheter, it could be an intravenous catheter for giving drugs or blood transfusion. So a biofilm is a microaggregation of bacteria. And these ones usually form a layer on these catheters. So you know that there's an infection going on. And it's important to know after how many days should you change a indwelling a urethral catheter or an intravenous catheter, so that you do not have bacteria attaching onto the catheter and getting into the body and causing very serious infections. So these are diagrams showing to the left solid artificial media or culture media. To the right is liquid medium, which referred to as broth. So on the left, the original culture medium is pink in color. Then you see these growth. These are what we refer to as colonies. And when we are describing the colonies, we refer to it as colonial morphology. So when you're describing colonial morphology, you want to know, is it smooth? So you describe the color, describe the size, is it large or small? Of course, you have to have seen several colonies for you to be able to say this is small in comparison to what. Is it smooth? This one appears smooth and shiny. So for bacteria that have capsules, they have a very mucoid. They appear mucoid. They form mucoid colonies. Some are very rough, some are very large, some are very small. To the right are two broth media. The right one is clear, so you always hold it against the light so you can see through, so you know that's clear. There's no bacterial growth. But on the one to the left, there is turbidity, so you cannot see through it, which means that bacterial growth has occurred. So the other thing that we describe is the bacterial growth phases. So we said bacteria increase both in size and in number. And we usually draw a graph showing the time and the different stages of bacterial growth. And the first one we refer to as the lag phase. And this is where the cells prepare for growth. So there is no increase in number of the bacteria, but there's an increase in size, because this is where bacteria are taking in nutrients, they're getting larger, they're preparing to divide. Then you have the exponential phase, which is where there is a high rate of cell division. So increase in cell number at an exponential rate until the maximum is available for the available conditions. Remember that for the artificial media, you can only have so many nutrients in that. The first one is a petri dish where we put the solid media, and the tube where we have the liquid media. So they will grow up to a point where there isn't any more available nutrients. Then they get into a stationary phase, which we also refer to as a post-exponential phase. So the total bacterial cell number reaches a maximum and stabilizes. So what happens here is that we do not have, the nutrients are now reducing, and we have the stage 4, which is the decline. We also refer to it as the death phase. It is also at this point that the bacteria that form spores and start forming spores because they do not have enough nutrition. So they need to survive and so get in the spore formation stage. So this is the graph that we use to describe the bacterial cell growth. So it's a log number of bacteria with time. We have the lag phase, the logarithmic or the exponential growth phase. We have the stationary phase. You have the death of the logarithmic decline phase. So how do we grow bacteria in the lab? We said it's very important to isolate the bacteria that's causing different organisms. You'll be able to, one, it's important for surveillance. You want to know that this pneumonia is an outbreak of pneumonia. It's being caused by an outbreak is being caused by a certain bacterium, which is why it's important to grow the bacteria in the lab. This is also the basis of the practicals that you're going to be doing in bacteriology. They'll be revolving around the growth of bacteria in the lab. So solid media, you'll be seeing solid media a lot. Solid media is, we make it and put it in a petri dish. So this round thing is a petri dish. You have an artificial media there, which is that color. The basic media is also referred to as a simple media. Examples of these are nutrient agar, which you'll find us abbreviating a lot as Na, which is solid. Then we have the nutrient broth, which is liquid. So it's also a basic medium, but it's liquid. So basic media supports the growth of microorganisms, but they do not have special nutritional requirements. Then we have the enriched media, which is basic media which can be enriched with different things. You can have hololyzed blood, you can have serum, you can have peptones, yeast extracts, vitamins, and other growth factors. One of the enriched media that you're going to be hearing of a lot is a blood agar. I'm going to hear a lot of blood agar. So remember, blood agar is an enriched medium, enriched with hololyzed blood, which is important, horse or sheep blood. So we use horse or sheep blood. So it's important for the growth of many medically important bacteria. Then we have enrichment media. So do not confuse enriched media with enrichment media. Enrichment media is a fluid, it's a broth, it's a fluid selective media. It contains substances that inhibit the growth of unwanted organisms. It's usually used where you get a specimen from an area which is naturally very rich in normal flora. So for example, examples of enrichment media is alkaline peptone water. This is an important enrichment medium for the causative form of cholera, which is Vibrio cholerae. So you get a specimen from the patient, you do not take it straight to a solid medium. You take it to the enrichment medium, get good numbers of the bacteria if they are present, and then we call it inoculation. Inoculate it onto the solid medium. And we have selective media. Now these are usually solid media. And they're called selective because they contain substances. It could be bile salts, it could be chemicals, it could be antibiotics. Now these substances inhibit the growth of one organism to allow the growth of another. An example is TCBS. So TCBS is a selective medium for the causative form of cholera, again, Vibrio cholerae. So that's what, it's green in color originally. So if you have The bacteria that cause cholera, they're going to be yellow in color. So these are colonies formed by Vibrio cholerae. Then we have differential media, also referred to as indicator media. And this is media to which dyes or other substances are added to differentiate microorganisms. An example is to your right. So we have a petri dish containing an indicator medium, which is a McConkey agar. You're going to be hearing a lot about the McConkey agar in bacteriology. So McConkey agar divides bacteria into two because it has lactose. So there are those that ferment lactose, so they will appear pink on McConkey. There are those that do not ferment lactose, so there's no change in color. So it's very important in helping us to differentiate or to classify bacteria into two. This is especially the bacteria that cause infections in the gastrointestinal tract. So lactose fermenters and those that do not ferment lactose, the non-lactose fermenters. And then you have transport media, which is semi-solid media. Transport media is important, especially if you're working in a place without good lab support. You need to get a specimen from the patient and transport it to the lab, which is some distance away. Bacteria can die very easily when they're outside the body. So you need to keep them alive long enough for them to get to the lab and inoculated and isolated. So we use transport media. Now, transport media contain ingredients to prevent the overgrowth of commensals. Commensals are normal flora. Because you're interested in the pathogenic bacteria, you're not interested in the normal flora. An example is Stewart's media. You can see a photograph on the right. Stewart's media is important for specimen coming from the genital tract, especially when we suspect Neisseria gonorrhea, which is a causative form of gonorrhea. So it ensures the survival of aerobic. If you suspect an aerobic organism, then you'll use a transport media which supports aerobes. If it's anaerobic, then you'll use transport media that supports anaerobes. So that once they get to the lab, then culture is done after collection, immediately when the specimen gets to the lab. Then you have identification media. And this is media to which substrates or chemicals are added to help identify bacteria isolated on primary cultures. Primary cultures is usually the very first growth. So you have a specimen, for example, you have a patient with pneumonia. You take a sputum sample and you put it, we call that inoculation, you inoculate it onto a solid medium. That's called a primary culture. If that bacterium forms colonies, that's a primary culture. The second step in the lab of laboratory diagnosis is usually to identify. So you have a growth, but which organism is it? So a good example of identification media is TSI, the triple sugar ion, which you'll hear a lot when you're looking at bacteria, which are important causative forms of gastrointestinal tract infections. So organisms are usually identified by changing color. If you look at the four, those are four different. The original color is the same. The original color is pink, the one at the extreme right. So if you have different bacteria, they give different reactions depending on what you have isolated from the patient. So it can either be yellow, it can be yellow and pink. It can be black, which means that this organism is producing hydrogen sulfide gas, or it can just be pink in color. So you very quickly are able to identify by changing color, but also production of gas. For example, in the first one on the left, there's a space. So the original one does not have a space between the medium and the bottom of the glass. So this space means that that bacterium produced gas. And also the one that causing blackening of that tube or that medium, it shows that it's producing hydrogen sulfide, which is a very important identification feature of one of the important organisms. So remember, we talked about the growth atmosphere. So we have aerobes and you have anaerobes. So how do you achieve these in the lab? So for aerobes, which are able to survive in presence of oxygen, as well as facultative anaerobes, which also survive in presence of oxygen, we put, once you've inoculated this onto the agar, we say we culture in air. So they're able to survive in air. However, for the strict anaerobes, they require complete absence of air. You have to achieve anaerobiosis in the lab. How do you achieve complete absence of oxygen in the lab? We have what we refer to as the anaerobic jar or the Fields Macintosh jar. Then we have those that require decreased levels of oxygen or increased levels of carbon dioxide. We use the candle extinction jar where we do not have the carbon dioxide incubator. And for temperature, remember we said most of these are mesophiles. They require a growth temperature of between 35 to 37. Temperature is achieved using an incubator. So these are diagrams showing a Fields Macintosh jar. During your practicals, you're going to be told the different parts of the Fields Macintosh jar and how anaerobiosis is achieved using the jar. We have the candle extinction jar, simply a jar and a candle in cases where you do not have the carbon dioxide incubator. Okay, so that is the end of introduction to bacteriology, bacterial anatomy, and bacterial physiology. Are there any questions? So any questions at all? See, someone is asking to share the slides. Yeah, we will share the slides. So I think if there are no questions, I don't see any in the chat, we can call it a day.